In genetics and biochemistry, sequencing means to determine the primary structure of an unbranched biopolymer. Sequencing results in a symbolic linear depiction known as a sequence, which succinctly summarizes much of the atomic level structure of the sequenced molecule. DNA sequencing. DNA sequencing is the process of determining the nucleotide order of a given DNA fragment. So far, most DNA sequencing has been performed using the chain termination method developed by Frederick Sanger. This technique uses sequence-specific termination of a DNA synthesis reaction using modified nucleotide substrates. However, new sequencing technologies such as pyrosequencing are gaining an increasing share of the sequencing market. More genome data are now being produced by pyrosequencing than Sanger DNA sequencing. Pyrosequencing has enabled rapid genome sequencing. Bacterial genomes can be sequenced in a single run with several times coverage with this technique. This technique was also used to sequence the genome of James Watson recently. The sequence of DNA encodes the necessary information for living things to survive and reproduce. Determining the sequence is therefore useful in fundamental research into why and how organisms live, as well as in applied subjects. Because of the key importance DNA has to living things, knowledge of DNA sequences are useful in practically any area of biological research. For example, in medicine it can be used to identify, diagnose, and potentially develop treatments for genetic diseases. Similarly, research into pathogens may lead to treatments for contagious diseases. Biotechnology is a burgeoning discipline, with the potential for many useful products and services. The Carlson curve is a term coined by The Economist to describe the biotechnological equivalent of Moore's law, and is named after author Rob Carlson. Carlson accurately predicted the doubling time of DNA sequencing technologies would be at least as fast as Moore's law. Carlson curves illustrate the rapid decreases in cost and increases in performance of a variety of technologies, including DNA sequencing, DNA synthesis, and a range of physical and computational tools used in protein expression and in determining protein structures. Sanger sequencing in chain terminator sequencing. Extension is initiated at a specific site on the template DNA by using a short oligonucleotide primer complementary to the template at that region. The oligonucleotide primer is extended using a DNA polymerase, an enzyme that replicates DNA. Included with the primer and DNA polymerase of the 4-deoxynucleotide bases, along with a low concentration of a chain-terminating nucleotide. Limited incorporation of the chain-terminating nucleotide by the DNA polymerase results in a series of related DNA fragments that are terminated only at positions where the particular nucleotide is used. The fragments are then size-separated by electrophoresis in a slab polyacrylamide gel, or more commonly now, in a narrow glass tube filled with a viscous polymer. An alternative to the labeling of the primer is to label the terminators instead, commonly called diterminator sequencing. The major advantage of this approach is the complete sequencing set can be performed in a single reaction, rather than the four needed with the labeled primer approach. This is accomplished by labeling each of the deoxynucleotide chain terminators with a separate fluorescent dye, which fluoresces at a different wavelength. This method is easier and quicker than the dye-primer approach, but may produce more uneven data peaks, due to a template-dependent difference in the incorporation of the large dye chain terminators. This problem has been significantly reduced with the introduction of new enzymes forms and dyes that minimize incorporation variability. This method is now used for the vast majority of sequencing reactions as it is both simpler and cheaper. The major reason for this is that the primers do not have to be separately labeled, although this is less of a concern with frequently used universal primers. This is changing rapidly due to the increasing cost-effectiveness of second and third generation systems from Illumina 454 AB Helicos and Dover. Biosequencing, which was developed by Pal Nyreq10 and Mustafa Runagi, has been commercialized by Biotage and 454 Life Sciences. The latter platform sequences roughly 100 megabases in a 7-hour run with a single machine. In the array-based method, single-stranded DNA is annealed to beads and amplified via EMPCR. These DNA-bound beads are then placed into wells on a fiber optic chip along with enzymes which produce light in the presence of ATP. When free nucleotides are washed over this chip, light is produced as ATP is generated when nucleotides join with the complementary base pairs. Addition of one nucleotide results in a reaction that generates a light signal that is recorded by the CCD camera in the instrument. The signal strength is proportional to the number of nucleotides. For example, homopolymer stretches incorporated in a single nucleotide flow. True single-molecule sequencing, large-scale sequencing, whereas the methods above describe various sequencing methods. Separate related terms are used when a large portion of a genome is sequenced. Several platforms were developed to perform exome sequencing, whole genome sequencing. RNA sequencing. RNA is less stable in the cell, and also more prone to nuclease attack experimentally. As RNA is generated by transcription from DNA, the information is already present in the cell's DNA. However, it is sometimes desirable to sequence RNA molecules. While sequencing DNA gives a genetic profile of an organism, sequencing RNA reflects only the sequences that are actively expressed in the cells. To sequence RNA, the usual method is first to reverse transcribe the RNA extracted from the sample to generate cDNA fragments. This can then be sequenced as described above. The bulk of RNA expressed in cells are ribosomal RNAs or small RNAs, detrimental for cellular translation, but often not the focus of a study. This fraction can fortunately be removed in vitro. However, to enrich for the messenger RNA also included that usually is of interest, derived from the exons. These mRNAs are to be later translated to proteins that support particular cellular functions. The expression profile therefore indicates cellular activity particularly desired in the studies of diseases, cellular behavior, responses to reagents or stimuli. Eukaryotic RNA molecules are not necessarily ecolinear with the DNA template, as introns are excised. This gives a certain complexity to map the read sequences back to the genome and thereby identify their origin. For more information on the capabilities of next-generation sequencing applied to whole transcriptomes, see RNA-seq and microRNA sequencing. Protein sequencing. Methods for performing protein sequencing include Edman degradation, peptide mass fingerprinting, mass spectrometry, protease digests. If the gene encoding the protein is known, it is currently much easier to sequence the DNA and infer the protein sequence. Determining part of a protein's amino acid sequence by one of the above methods may be sufficient to identify a clone carrying this gene. Polysaccharide sequencing. Though polysaccharides are also biopolymers, it is not so common to talk of sequencing a polysaccharide for several reasons. Although many polysaccharides are linear, many have branches. Many different units can be used and bonded in different ways. However, the main theoretical reason is that whereas the other polymers listed here are primarily generated in a template-dependent manner by one process of enzyme, each individual join in a polysaccharide may be formed by a different enzyme. In many cases, the assembly is not uniquely specified. Depending on which enzyme acts, one of several different units may be incorporated. This can lead to a family of similar molecules being formed. This is particularly true for plant polysaccharides. Methods for the structure determination of oligosaccharides and polysaccharides include NMR spectroscopy and methylation analysis.